think we'll get started. Good morning. It's my pleasure to introduce our Grand Round speaker today, Dr. Yasmin Hurd. Dr. Hurd is the Ward-Coleman Chair in Translational Neuroscience, as well as Professor of Psychiatry, Neuroscience, and Pharmacological Sciences at the Icahn School of Medicine. She is also the director of the Addiction Institute at the Mount Sinai Behavioral Health System. She is an internationally renowned neuroscientist whose preclinical and clinical translational research examines the neurobiology of substance use disorders and related psychiatric disorders, with a primary focus on opioid abuse and the developmental effects of cannabis. Based on her high scientific accomplishments, she was inducted into both the National Academy of Sciences and the National Academy of Medicine. Please join me in welcoming Dr. Hurd.
Thank you very much. It's great also being here and seeing some medicine friends that I haven't seen in a very long time. Clearly, we need to have more interdepartmental events. So today I'm going to talk about cannabis, and obviously in part the presentation is going to be biased towards the brain, but it's really like, how do we come from a drug to a medicine? And everybody thinks about cannabis, and today, at least in my world, people frame cannabis as being this wonder drug now, basically can cure everything. I think that I don't know if there's any other quote unquote drug in history or now medicine that has been given this wide range of effects that they think that it can cure, can treat, whatever word you want to use. I remember one was even, there was something even, well, a lot of skin conditions, a lot of medical issues, psychiatric issues, everything. But the question is, what is cannabis? And when you look at cannabis, it really is a very complex plant. It has over 500 chemicals, over 140 of them are cannabinoids. We know mainly THC, or I should say we study THC the most, because that is the cannabinoid that is associated with the rewarding effects of the drug. But as said, it contains all these other hundreds of chemicals, other cannabinoids and terpenes that we still have very little clue about what they do. So when we also think about cannabis in terms of the complexity, it's also the fact that it is a very changing product. And cannabis today is very different from the cannabis that was on the planet to start. When we think about THC in particular, the cannabinoid, as I said, that most people really focus on when you think about cannabis, the concentration of THC in the plant originally was about 2 to 4%. Today, it's astronomical, going even to over 15%. And another cannabinoid that I'll talk about more near the end of my lecture, cannabidiol, CBD, that used to have a much higher percentage in the cannabis plant. And the ratio between THC and CBD today is, as compared to the original plant, is quite distorted. And you'll see why that makes a big difference. The cannabis today also, not just the plant and the THC concentration, the variety of products that are available are, again, astronomical. And there are varied routes of administration. So dabbing can even give THC concentrations on these concentrated cannabis THC products can even give concentrations of THC of the 90% that people are consuming. So think of 90% compared to the original plant, 2%. Another problem that we have is that, one is that cannabis legalization in the country is quite complicated as well. So federally, it's still illegal. But in 2018, the Congress passed the Farm Act, which made cannabinoids derived from the hemp plant, which is basically CBD, because that's a non-intoxicating cannabinoid. They made that legal. And so what that did actually was to make the cannabis industry explode. And it exploded by taking CBD and converting it chemically into intoxicating THC analogs. And when they convert it, the chemicals even that they use can have toxic effects. So when they say these light cannabis, like Delta 8, Delta 10 THC, they're not really quote unquote light. Another thing, as technology has improved and we were able to take the cannabis plant and look at, as I said, a lot of the chemicals that are in this plant, they're cannabinoids that are really low in concentration that we would never have been able to detect before these high mass spec analysis. And now people are making them synthetically, and they even have like 30-fold more potency than the Delta 9 THC. So this is the world that we live in terms of the increased potency of THC, and throughout the country, we see that as the THC potency has increased, so has the increase of cannabis use, and that has a huge issue because as the potency of the most intoxicating component of anything increases, so does the addiction vulnerability. Today in the U.S., more people consume daily or near daily cannabis than they do alcohol. So that again is one of the major issues that we're facing. So I'm going to give you just a little bit of overview of some of the consequences of cannabis use. Obviously, recreationally, people take it to feel intoxicated, the euphoria. However, obviously, it has short-term memory issues. It also produces, motor coordination issues. But the question is like, not when someone goes to a party necessarily, the same thing with the alcohol, take a couple of beers or wine. It's more about what happens during the long-term use. And so as a long-term use, we see definitely issues of, sleep impairment, more memory issues and learning problems, but also then starts to lead to addiction and the craving for the drug. Yes, many people would, say it, long-term, it decreases appetite, even though it might acutely increase like the munchies or so on. But absolutely, there are long-term changes in the brain related to like aggression, irritability, especially during the withdrawal. But it's not just the mental issues with cannabis. There are also physical effects that we see with chronic use. The one good thing perhaps in terms of the cannabis industry exploding is that they're not just smoking anymore. There are various ways of taking cannabis, but we do know and see that smoking, just like cigarettes, does increase like bronchitis, chronic cough. Many people don't realize a lot of the heart rate changes, blood pressure changes. We also see now more myocardial infarction, especially in men, young men consuming cannabis. We see also changes in inflammation I'll come back to. But one of the things that your ED colleagues will tell you, so many more people showing up in the emergency room with the cannabinoid hyperemesis syndrome. Basically, this vomiting, nausea, abdominal pain. In pregnancy, you'll have pregnant women who consume cannabis and have a cannabis use disorder, and you can easily tell the difference between pregnancy-induced hyperemesis, because if they stop using cannabis for a few days, that the nausea and all of those things go away. Also, they get very hot, and they have to take a lot of their body temperature changes a lot, so they take all these very hot showers. So very different, and we never had these things showing up in the emergency room before. But I'm going to come back to the fact that many people think that cannabis use, you can't have an addiction, but the DSM diagnostic manual, not just in terms of the use disorder and the characteristics that are part of that diagnosis, but really cannabis use disorder, the withdrawal is very significant. And I will get emails from people even asking me, even last week, 2 weeks ago, I had Dr. Hurd, I have a cannabis use disorder. I've been really trying. It's the withdrawal that gets me. Can you help? Because the amount of, like we see all these sleep disturbances, the mood effects, it's really intense. So that's the complexity of it, but of the cannabis plant, but it's impacting on our endogenous biological systems. And even that is very complex. So you have to understand the endocannabinoid system. So the endocannabinoid system consists of these endogenous cannabinoid ligands that many people think, oh, cannabis has been around for thousands of years, so, it's natural, or we have an endogenous cannabinoid system. And it's true, we do, and we have these different ligands. But this is actually a relatively recent discovery in terms of understanding even these endocannabinoids, for example, anandamide, AEA, and 2-arachidonyl glycerol, 2-AG, that was in the 1990s. So we're still learning a lot about these. These are lipids, they're made on demand often, and they interact with the endogenous cannabinoid receptors, which are the CB1 receptors and CB2, and I'm gonna come back to them in a little bit. But what we now know is that these two endocannabinoids that people study a lot and think, okay, these are two endocannabinoids, it really isn't. These two endocannabinoids belong to this huge, diverse, and actually I'm not even showing the vast majority of it, lipid biological processes in the body. And we have very little understanding of these lipids. They're biologically active, so you're saying you're changing one aspect of an endocannabinoid ligand, but you're changing a whole biological process. So another thing about the endocannabinoid system is that they're actually modulators of most biological functions. So the schematic on the left reflects the cannabinoid receptors. There are mainly two cannabinoid receptors, the CB1 and CB2. You can see they're expressed throughout the body. I'm biased, I will focus on the brain, and the brain definitely has a high abundance of cannabinoid receptors. But they play a really key modulatory role. In addition to the cannabinoid receptors, there are other receptors that we're still learning about that mediates the actions of our endocannabinoids. And the enzymes that synthesize and break down these endocannabinoids are being targeted for medicinal development. But again, we do not understand the full complexity of them. And because the cannabinoid receptors and the endocannabinoid systems are expressed in basically every organ, they are critical for all of these biological functions. And that is, again, one of the issues that we have with how the cannabis plant has changed and the potency of cannabis that impacts on the endogenous cannabinoid system. So like I said, I'm biased. So I'm biased for the brain, and I'm biased for the brain in regard to cannabis because the cannabinoid receptor, the CB1 receptor, is the most abundant G protein coupled receptor in the brain. It is critical for regulating synaptic function in terms of modulating cell communication. The cannabinoid receptors are on the presynaptic terminals that then if something is too high, if a transmitter release, especially the excitatory, it will start to shut it down. And if the inhibitory transmitter is also too high, it also goes and mediates a balance. So just like in other organs, the endocannabinoid system is about regulation and keeping things in homeostasis. So this critical biological process for me, and especially in the brain, is important when we think about psychiatry or psychiatric risk. And this number just reminds me, again, to emphasize that many people think that you can't develop a cannabis use disorder, but we know about 10 to 30% of people will develop a cannabis use disorder with cannabis use, which is actually similar to other substance use like heroin or so on, cocaine. However, the reason why you have the similar number of cannabis use disorder is that two things. One, the prevalence of cannabis is much higher than, for example, cocaine use in our society. And today we see that the THC concentration, as I said, is getting higher. Another thing that we know in terms of cannabis use disorder is when we study the functional changes in the brain and the structural changes in the brain, mainly from neuroimaging studies or so on, we see that the changes in the brain are very similar to individuals who might have an opiate use disorder, cocaine use disorder. So those brain regions, I don't remember if many of you remember all of your neuroscience, so I won't go into all of the specific brain regions, but to say you see very similar things that are happening. So it's not just cannabis use disorder that we in psychiatry have an issue with. The problem is that we see a relationship to schizophrenia and all these psychosis, depression, anxiety is huge. All the patients will tell you that they're using cannabis to alleviate their anxiety, but every data for us shows that it exacerbates it. And cannabis use disorder has a high psychiatric comorbidity with other disorders in terms of, again, the anxiety and schizotypical disorders. But perhaps one of the most important thing in terms of why I went down this path with studying cannabis and brain health is that we now know that, even though most psychiatric illnesses are diagnosed in adulthood, we now know that they have a developmental origin. The vast majority of psychiatric disorders. We're never studying early life. We usually study the person that's in front of you. So of course, adolescence is a critical window for when, teens will experiment with cannabis, but also we know that psychiatric illnesses even have an in utero early developmental origin. So why this is critical is that the endocannabinoid system is actually essential for neurodevelopment. It's important, it helps the cells, new cells to be born, the migration of those cells that form the synapses. In fact, it's critical for hard wiring of the brain. And throughout your early life and even into adolescence and young adulthood, it helps to fine tune the maturation of for example, the prefrontal cortex, but every brain region, to be honest. So what we see today is that more pregnant women are consuming cannabis, more kids actually, because when different states have legalized cannabis and more kids indoor are experiencing secondhand exposure. And of course, during adolescence, as usual, we have a much larger percentage of kids today consuming cannabis. So I'm going to talk to you about a few of those developmental windows and what it may mean. And you can think about it in terms of the individuals and patients who you treat. As I mentioned, endocannabinoid system critical for neurodevelopment. The picture on the bottom was just showing the expression of the cannabinoid receptor in the human fetal brain. And we could see that unlike the adult brain, the cannabinoid receptor is highly expressed in brain areas we call the limbic brain areas, important for emotional regulation and so on. And the endocannabinoid system is very dynamic in terms of the enzymes that synthesize and break down these endogenous ligands. So we've studied the human fetal brain. I'm not going to just give a summary that have been exposed in utero to cannabis. And we can see genes and proteins that are relevant to what I said before, what we know, the cytoskeletal organization of the brain. For example, that the fetuses show with cannabis exposure show changes in these proteins and these genes relevant to endocannabinoid system being important for hardwiring of the brain. We know it changes our endogenous opioid system. Endogenous opioid system is critical for many things, not just reward and motivation, but even pain and stress responsivity. And it even correlates to the amount of cannabis the mothers use. In addiction and many psychiatric disorders, we talk about dopamine, that neurotransmitter that's really important for saliency reward. And we see here, even in the mid-gestational fetal brain, that those that were exposed in utero to cannabis show this really significant change in the brain region of the nucleus accumbens, again, important for reward motivated behavior. Also in a brain region called the amygdala, where we saw that the males were showing the greatest changes. Importantly, we can replicate this in our animal models. So we know that THC is inducing the changes that we see in the human fetal brain. But the question isn't whether or not exposure is, these changes in the brain, are they relevant to the phenotype that you see later? So I'm masochistic, but I have a masochistic colleague, an even more masochistic colleague, Yoko Nomura. And Yoko was studying stress during pregnancy and looking at the kids to see whether or not they may show different behavioral events that may predict aspects of psychiatric vulnerability later in life. As I said, we now understand that the developing brain is critical for later psychiatric risk. And I wanted to look at substances as cannabis. But every pregnant person will have different stressors. But one thing happened in New York about a little over a decade ago was Hurricane Sandy. And that was the objective stress we used. And we had people in before Sandy, during Sandy, and post Sandy. And the question I want to know is, can we have some clue about what happened in utero? And so we decided to study the placenta, the most critical temporary organ. You guys all know about it. I don't have to tell you what the placenta is, but we do know, interestingly for psychiatric illnesses, but also physical illnesses and many disease vulnerability, that the placental program is essential for that. And so we sequenced the placenta, and we could see, for example, the cannabinoid receptor was changed, and it correlated to the amount of cannabis the mothers reported using. But we sequenced because we wanted to look in an unbiased manner. And what we saw that was really changed was the immune system is very strongly changed in the placenta of the women who used cannabis. No matter how we looked at it, the immune system was changed in large part. There was also a sex difference, and it was mainly, the yellow indicates the immune-related genes. It's the male placenta that's changed the most. Again, people can have different lives, so we use our animal model to see whether or not this is causal. So here the animals do vape, and so they vape, and once again, the placenta of the male placenta shows the most dramatic change in our animal models, similar to what we saw in humans. And animals, they will have multiple pups. And each pup has its own placenta, and that your sibling beside you may be female, yet still your placenta shows this strong immune dysregulation. We know that the endocannabinoid system is tightly linked to the immune system, especially for example, the CB2 receptor is highly expressed in immune cells. And it's very complicated, and we're still trying to understand, under certain conditions, we can see that there are anti-inflammatory effects of cannabinoids, but also there can be, and in specific cannabinoid, specific immune-related changes, and we can see that they're pro-inflammatory effects. So again, still do not understand the cannabinoids and cannabis very much, but we know that they play a very strong role, and as I said, tight regulation. So what about the phenotype of the kids? So we bring kids in practically every year till we had a little few years gap between a grant. And they go through clinical assessments. So for example, when you look at the hair, the cortisol stress hormone in the hair of these kids, you can see that those kids who were exposed to prenatally to cannabis, they show greater cortisol levels. They even show greater stress reactivity if you measure cortisol in their saliva. But the clinical measures, we saw that these kids showed greater anxiety, greater aggression, greater hyperactivity. But an interesting thing that you also see is that there is a synergistic effect. So those that were exposed to where the mothers had excessive stress and cannabis use, they're the ones that show a much greater effect, whether in anxiety, aggression, and so on, and even the age when they're diagnosed. So it's important to realize that things are obviously we're all complex, and so understanding the full pattern, it's not just the drug, it's also the environment and other environmental things that can synergistically interact to exacerbate certain disorders. But I mentioned we sequenced the placenta, and does the placenta gene expression pattern have anything to do with behavior, especially behavior long-term? And so doing a lot of computational type of work, we could see that the transcriptome of the placenta, but the immune transcriptome predicted the kids' anxiety behavior, for example, later in life. We're still following up with the kids. They're now about like 10 to 13, so they're coming back in, and so we're going to look and see if this continues throughout their lives. As I said, now this age window is very critical for a lot of the psychiatric vulnerability, and can we intervene to prevent the trajectory that we expect? I said it was masochistic, but I was studying animal models, looking at prenatal to adulthood, but our human studies actually told us we used to look only in adulthood, but we can see in the kids that there are things happening early on. So now we have our animal models and we're studying them at different windows. I'm not going to show you those, but just to say we see a lot of different changes on behavior, from motivated changes, depression-like changes, anxiety, stress reactivity changes in our animal models as well. So what is it that is maintaining these changes long-term throughout life from, that it's in utero exposure that can change your health risk? And so this is where we started looking at epigenetic mechanisms. And epigenetic mechanisms, because we know that we have not changed the DNA sequence of these kids, but the environment can change how genes that should be off are now turned on, and vice versa, genes that should be open are now turned off. There are now numerous epigenetic mechanisms in terms of the tags that are put onto DNA and how, like I said, DNA that's opened and closed. We're still learning a lot. But one of the things that we did was to probe for, just in an unbiased manner, the different epigenetic modifications that we knew at that time. And one of the things that we saw in the cohorts of this is our animal model, replicated also in our human, but we could see that a particular epigenetic tag, it used to be called mixed lineage leukemia, because in epigenetics, cancer field has really, we studied the cancer field because they have really characterized and helped us understand epigenetic mechanisms much more, but it's now called KMT2A, which is a histone methyltransferase. And basically, it's really an important epigenetic mark for transcriptional memory. And we can see that in the adult brains, but also in early development. For example, this nucleus accumbens region, like I said, important for reward, motivated behavior. We can see that this epigenetic mark is changed not only on the gene expression level, but also on the protein level, this increase. The one thing our animal models allow us to do, we can go into the brain, we can infuse a virus that can then reduce the expression of this epigenetic mark, and we can change their behavior. For example, adults, like I said, with prenatal THC exposure, they show changes in motivated behavior. And if you knock down this particular epigenetic tag, you can normalize their behavior. Clearly, we're not going to do that with kids, but we know that there's this mechanistic relationship. But when they get into adulthood and we look into the nucleus accumbens, a really interesting thing we see again. Immune-related genes in the brain, so where this epigenetic tag is binding, again, the yellow emphasizing the immune-related genes, there's a huge change in them. And where this epigenetic tag, you can see in those adults with only prenatal THC, there's a complete reorganization of where this epigenetic tag of the genes that it's now regulating, and again, immune-related changes. So that's prenatal. We know, like I said, during adolescence is the time when a lot of people will experiment. And we know also that this is a time period also of vulnerability for the developing brain. The importance of adolescence for us is that we do know that most adults with obviously a substance use disorder start in adolescence, so that's why we try to get earlier. But many teens actually have a cannabis use disorder. So this is one of the things for us in terms of trying to target. When you do human studies, neuroimaging, we see structural changes in the brains of those adolescents and young adults with cannabis use. And this is a study in Europe, Imaging, where they were imaging kids like every 2 years from, I think their study started around age 12. In the US, we have an ABCD study that started when they're at age 9. And you can see that these changes in cortical thinning in kids, or actually in kids over time that have used cannabis, and this cortical thinning actually like even correlates to their, not also their cannabis use, but their impulsivity, their decision-making behavior. But I will tell you, these human studies, there are a lot of debates because the genetics people talk about, their life environment. So that's why, again, we come back to our animal models. And the prefrontal cortex is the last brain region to really reach full maturity. And we can go in and look at these prefrontal cortical cells. And when we do in adults with adolescent THC exposure, not everything has changed, but what has changed is the complexity of these cells. In terms of the branches that they will make in terms of connecting with other neurons and other cells. But when the changes that we see on a morphological structure level, interestingly, looks like they're like stress models. And we can pick up these cells. We use laser, capture mic, and we can pick these cells up and we can sequence them. And one of the things that we saw, we were really surprised because there was a complete reorganization of the gene network in the prefrontal cortex of these animals. And what was changed was, one, related to structural organization and how synapses communicate with each other. So not surprising, we saw these structural changes. But what was changed even more dramatically were these epigenetic changes. And it was even the same epigenetic mark that we had seen in the prenatal exposure. And there was, again, this reorganization of those animals, of the epigenetic mechanisms here is like synaptic plasticity related genes. And when we looked in the brains of individuals that are diagnosed with different psychiatric disorders, the changes match those a lot that you see in individuals with schizophrenia. So we know that the endocannabinoid system, like I said, is impacting on the synaptic communication. But when we started these studies, we were looking at THC levels similar to what people had been using back then, which was about 4% THC. And the potency matters. The endocannabinoid system, as I said to you earlier, is critical for homeostasis. These ligands are very low in concentration. And now we have potency up to, like I said, even 90% THC. So you're changing the state of how cells are communicating. And people always say, oh, the teens will titrate the amount of cannabis they're consuming, so it doesn't matter that the concentrations have increased. Even adults don't titrate. So because it's not that they have other options. This is the THC today. And we know that the higher the potency, as I said earlier, the greater we see for addiction risk as well as psychiatric risk. So actually, Jackie Ferlin was a postdoc on my team. She's now assistant professor. We want to know, does dose matter? And so when we give animals low dose, which is now considered low dose THC, and high dose, we see their cortisol levels change. And it lasts, the high dose animals, and the cortisol stress levels are high for a very long time. If you give them a little stress, even just a little isolated stress, and you give them a reward, those animals that had high dose THC during adolescence, they're much more sensitive to the stress. We can look at changes in different brain regions, and we see significant changes. I'm not going to go into the details with them, but interestingly, they relate to impulsivity and decision making, but again, mainly when stressed. We even have animals self-administer THC. So now the one things that we see edibles are used, so our rats also get edibles, and they can control how much edibles they consume. And animals, just like humans, they will segregate, and some will really like it, and others won't. And we do a cognitive task with our animals, the same as with the humans. It's a decision-making task, and we look at different types of risky behavior versus optimal behavior. And those animals that tend to like the THC, the high consumers, they show much greater risky behavior. And that's what we see in our humans who have cannabis use disorder. They show much more risky behavior on these cognitive tasks. But once again, when the animals are stressed, their THC concentration, consumption goes up. So the stress is key. We're studying different brain regions. Here is a amygdala region, like I showed you before, important for actually emotional regulation. And what we saw that was changed a lot in the brain were astrocytes, for example. And astrocytes are critical regulators, even of the immune. They're part of the immune, the glial population. And they're also changed on a structural level as well. But again, only in those that were stressed and had adolescent THC exposure, and it related to their decision-making and impulsivity behavior. So stress, the endocannabinoid system, the immune system, they're all tightly integrated. And many people, as I said, the patients will tell us that they consume cannabis to reduce their stress. Short-term or low dose, yes. The endocannabinoid system is one of our brakes for helping us indeed manage stress. So it puts a brake, the endogenous cannabinoids, we get stress, our endocannabinoid system come into action, it helps to stop. But what happened with frequent cannabis users is that the levels of their cannabinoid receptors go down, so their natural brake on our natural stress system is no longer there. So that's one of the main issues. I put this here also to emphasize another aspect of cannabis use, the route of administration. So when animals vape, we can take their lungs and we look, and they have huge changes in their lungs, in the immune changes in lung. When the animals, obviously edibles, we don't see those changes. So vaping is a huge issue that we have to deal with, not just with the brain, as I said, in terms of we see greater addiction and psychiatric risk, but even in their lung capacity in the immune system. So this last part, I will just come to remind you that the complexity of the endocannabinoid system, but the complexity of the cannabinoids in the cannabis plant. I've talked to you mostly about THC when we do our animal models, but as I said, there are over 140 cannabinoids. And many years ago, I wanted to study another one because I thought we wouldn't see anything, or it would be similar perhaps even as THC, which was cannabidiol, CBD. And THC and CBD are very different. So that's another thing when patients talk to you about cannabis, what kind of cannabis are you consuming? Because, as I said, THC is associated with the intoxication, the reward. CBD does not. Anxiety, it's low dose THC that actually reduces anxiety. High dose does not. In fact, it exacerbates. CBD reduces it. And just even the side effects, THC has much greater side effects than CBD. CBD, we're learning a lot about. Unlike THC, that is much more, I think we know a lot in terms of it, the agonist of the cannabinoid receptors in large part. CBD has an action on multiple receptor systems, but very low. So perhaps that's why it has a low side effects. It doesn't have a high affinity at many of these biological systems. In fact, it's even a negative allosteric modulator of the cannabinoid receptor. It's not even an agonist. And I'm not going to go into, like, we're learning about what its main mechanism of action is. We started looking at cannabidiol for potential treatment of opioid use disorder, because clearly we're still in an opioid epidemic with a lot of death, overdose deaths. And it started with our animal model, because when we would give, for example, adolescent rats THC and study them for whether or not they would self-administer heroin in terms of, quote, not the gateway, but the gateway hypothesis, but more looking on the neurobiological level. They would always self-administer more, but like humans, some rats don't. But when we gave them CBD, we were surprised to see that instead of increasing their heroin self-administration, it actually decreased their heroin-seeking behavior. Because just like humans who have an addiction, its environmental context comes in a large part. So if you show them like a light cue or something in the environment when they get the drug, you just show them that and they're not getting the drug, that cue triggers their drug-seeking behavior. And that's what CBD was reducing. We studied the brains of people who, unfortunately, there are a lot of people who die from opioids, and we know the molecular changes that occur in their brain. And we can have our animal models and we can look and see these similar changes. And we could see that CBD normalized at least those synaptic related genes that we saw. So we decided to do a clinical trial to see, can CBD, it's not intoxicating, can CBD be used to treat opiate use disorder? And I'll tell you, since no one was studying CBD back then, I used the same doses that we gave to our rats. So, and now the whole field is using it. So just so you know. So our rats told us that it was seeking behavior. So we studied craving because that's why people really continue their substance use. And we showed them videos. And it was a double-blinded placebo control. And those individuals who had had a heroin use disorder, an abstinent. All those individuals who had placebo, and we showed them the heroin cue, they craved, and CBD reduced that. Our animal model also showed us like weeks later after their final CBD, it was still having an effect to reduce their heroin-seeking behavior. So we brought people back a week later, it was still reducing their craving. One thing our animals didn't tell us, because they don't speak, is that what our human patients don't realize, anxiety. So those who had saw the heroin cue but had gotten placebo, their anxiety went up, CBD reduced that. A week later after CBD, it was still reducing their anxiety. It's not only on a subjective measure, we also took cortisol level, and those individuals, when their cortisol levels went up when they had seen the drug cue and had gotten placebo, and CBD reduced that. Same thing with their heart rate, placebo, seeing the drug cue, their heart rate goes up, and CBD reduces that.
Good morning, how are you?
So now we're trying to understand the mechanism by which CBD is working. My postdoc, Alex Chisholm. So in our animal models, she replicated again, CBD reduces heroin-seeking behavior. But now we're also looking at anxiety-like behavior in our animals because that's what our patients tell us. And indeed, when you give an aversive cue to an animal and you show that to them and they get the placebo, you can say, their anxiety-like behavior goes up. If they get CBD, it reduces that. So the animal model replicates what we see in our animals. So now we're trying to understand, like I said, the molecular mechanisms. And we can see that genes are turned on and turned off in different brain areas. I'm showing you the accumbens here when they're with heroin. And CBD is changing that. CBD is changing it, it reverses it. It reverses many of the not many, it reverses specific biological processes, and some of them relate to neuroinflammatory processes. So the changes that heroin is inducing, and we also see changes in epigenetic mechanisms, and in the brains of opioid users, we also see these changes that actually reflect neurodegenerative processes, and CBD reduces that. And the genes that they reduce, or they switch and normalize, correlates to their heroin-seeking behavior. We now know that the same things are happening with anxiety-like behavior, and their shared biological systems that they're studying. So clearly, we think that cannabidiol does hold promise as a treatment for certain substance use disorder. Other people have replicated our studies with now looking at cannabis use disorders, tobacco, and so on. We still don't know dose, we still don't know the treatments, and clearly the formulations and the delivery system makes a difference. So we're doing neuroimaging studies. We have a study that should be ending in June, looking at what's happening in the brains of these individuals with CBD. And we're now starting phase 3 of looking at people with opioid use disorder with CBD, where they're in their natural environments, about 6 months with CBD. We just finished the phase 2. And we hope that we will indeed, that something will show to be positive as a treatment, but we're still double-blinded for even our phase 2. I don't know the results, except the statisticians did show we have individuals wear an Oura ring. Because it's not just the subjective effects, because people can tell us, oh, we think this is helping us. But in their natural environment, we can now track their physiology. And we see that those who had placebo, they spend more of their day in a stressful state. And those with the CBD, they spend less of it there and more in the quote unquote normal and restorative state. So we'll see if that replicates, but having these measures in the real world, I think, is really critical. So, are there risks and benefits? Is it a drug in terms of, recreational drug, or are there some medicinal benefits? And on both sides of this, this debate, cannabis can have risk and can have benefits. We see that there are aspects of dose. Age matters. I didn't tell you about geriatric people, but even that is important. But the condition matter. The drugs in a healthy individual versus those that have a medical condition definitely differs. The route of administration differs. And also, one of the things, the cannabinoid differs. So when people tell you they're consuming cannabis, they're not consuming cannabis. They're consuming 500 chemicals. If the chemical has more THC, we see more risk with it. If it has more CBD, we see less risk. But we still have a lot that we don't know because most outcomes for many of the disorders are still unclear. So with that, I thank my preclinical team, and we've also not enough space for all of the clinical, the CRCs, but I thank them all. They're amazing for our masochistic studies. Don't study development, and I'm happy to take any questions that you might have.
Thank you for that incredible talk. Are there any questions from the audience? Fiona?
Thanks, Yasmin. That's a cool talk.
Thank you.
I'm interested in the immune signature changes that you're seeing, obviously, in these mice, particularly in like the CBD cohort. Have you been able to separate, like, so you haven't done like this sorting, it's just follow a whole brain tissue?
Correct. And so now we're doing single cell sequencing. Well, we're doing single cell sequencing more first with the heroin, and then looking at it with the CBD. Now, when we saw that CBD was reversing a lot of this, we want to see in particular what cells that it's. But as I said, we expect a lot of astrocytes perhaps and microglia. We see huge lipid-related changes and linoleic acid related to even the anxiety that's changed. And we think that there is aspects, strong mitochondrial changes and the blood-brain barrier. So both with, many people don't realize that stress does eat away at your blood-brain barrier. And that's why I think perhaps the immune perturbations get in, and they are inducing these changes. So we're trying to see, so CBD is changing some of the endothelial related perturbations. So we're trying to see if that's enough to perhaps curb some of the immune, but I'm not an immunologist, which is challenging. So that's one of the things you're all, the same thing studying placenta. I had no clue, like, why do I go down these rabbit holes? So, but that's why I love science, right? You're always learning something new, but we need a lot of immunology help. Yeah.
I'm sorry, just to cap that, on the same lines, have you seen some of the, or have you looked at some of the effects of CBD on like Alzheimer's or a lot of these neurodegenerative models, especially since you're seeing the same sort of signatures in your animal model?
Yeah. I realized I didn't say the question before. So Fiona now asked, do we see changes in Alzheimer's models or so on with CBD? Because absolutely, I don't think that what we see in the human brain is Alzheimer's. It's in the same direction, but Alzheimer's is at one end. So we do see this neurodegenerative signature and the tauopathy type of effects. We're working with Fanny El-Hahi, for example, who's developed some of the tau measures and markers, and we're going to see if we can, if CBD, how CBD works in that model, but we haven't studied it in any Alzheimer's specific model as yet. I just think it will be too late. I think perhaps earlier. So again, prodromal, if you know that earlier you can start using, and I don't know if CBD is what the true thing of of Alzheimer's. But I can say that we also, in the human brain, we saw that the most epigenetically changed loci, the gene, was FIN, and FIN phosphorylates tau. And when we give a FIN inhibitor, which is being developed for Alzheimer's, we'll see how that goes, it actually reduces heroin self-administration and seeking behavior. So we're also working in that model to try to see if we can run clinical trials with that for opioid use disorder. We'll see. Yeah. Sorry. And back there too. Sorry. Hi, Monica.
Thanks for being here. I also want to thank you for the support, your support of the All of Us program as we start a new aspect of the protocol. We'll recruit participants with opioid use disorder. Really couldn't have done it without you. So thanks for that.
Thanks.
Great talk. I had a couple of questions about reversibility of some of the changes that you're seeing. I know that with the CBD, potentially you might, just even without intervention, are any of the epigenetic changes reversible over time in your animal model? And you may have covered that behind this.
Yeah, the question is, are any of the changes, especially in our animal model, because that's what we can look at, reversible? So a key thing is that we do see that many of these are epigenetically mediated. And by that, epigenetics means it's reversible. So we can reverse them with epigenetic drugs, you know, and so that again is like one aspect of clinical trial. A lot of companies don't want us to use their medication for addiction. So there's a stigma also, yes, which is bad. But I do think that a number of these are reversible to a point. And I think to Fiona's, you know, point about perhaps this neurodegenerative, it's not Alzheimer's, but if you pass a point. But I do think that they're reversible. And do you need medication? Perhaps not. And that's one of the things about early behavioral intervention, psychosocial, because you know, even our animals, social contact makes a huge difference. So there are things that perhaps we can do to reverse it because it is epigenetically being modulated.
Inflammatory signatures that you're seeing there that you think is reversible? It seems like it could be inflammation.
Yeah.
Seems like it would, but I don't know if you're seeing that.
I've not studied it because the immune system, I'm like, oh no. Yeah, so that is definitely one of the things in terms of we can, you know, what are some good anti-inflammatory medications that could be tried, and could those help? So yeah, definitely important to do. Yeah, thank you. Wait, what kind of exposure?
Secondhand exposure. And the other one is like if you have any like long-term data, like smoking, risk for COPD or like lung cancer.
Yeah, so my colleague Risat Sinai at NIH, so we are collaborating with him on like the lung things because again, I've no, like the lungs beautiful, but the brain is much more, yeah. So we see these changes, especially with the vaping model, and so people need to realize that when they're vaping, they think it's a healthier, quote unquote, route, but it's impacting the lung immune system a lot. So the question was also secondhand smoke. The CDC, when they were allowed to exist, I don't know what's going to happen, a few years ago, they're the ones who showed the data of secondhand smoke in kids, and secondhand cannabis smoke, I should say, in kids, and seeing greater COPD-related type of illness. I don't know how the data is today, but in our animal models, I would say that Resat, those are things that they are studying in terms of from the animal model part, you know, what's related to they're actually, it's not secondhand smoke, they're actually consuming the vaping. And then, like I said, we'll see whether or not their studies show that it can be reversed. But I don't really have any firsthand data so we don't do a secondhand model, but like I said, data from the CDC and from a few other colleagues with looking at kids, which is actually a challenge for us as well. I shouldn't say that. So our kids in our that we've been studying from, they were in utero, they are also being exposed to things in their environment now. So it's not just their prenatal. So we're trying to account for that and trying to see if we can, parents obviously don't tell us everything. So we're trying to see, do certain toxicology again in hair, just to try to get a sense if we can get that kind of information. But I don't really have that much right now for it, as they're just actually coming back. So, thanks. Thank you all. Thanks.